the fascia slash interstitium.
Got
And
it.
the new terminology that my team is working towards, and it's a group of very high level anatomists. We're not talking like a local sort of thing. These are international level conversations we're having, is that there's a fascial system that is pervaded by the fascial interstitium. Interstitium literally means the in-between. The interstitium that has, it's not a new term. There's been classically recognized the interstitium between some cells. Not all cells, most cells are tight together so that there's no hyaluronic acid or spaces between them. But the main part is around every capillary. There has to be a space around the capillaries before you get to the cells through which nutrients can enter the tissue to encounter the cells, and through which waste products from the cells can get into the vascular system for elimination. So that's the interstitium classically. What my team is responsible for is, no, there's an even larger scale, and this may be, in fact, a fractal network. And these interstitial spaces are
That's what I was
10 to
thinking about.
20 times the size of the pericapillary ones. So they're much larger scale. And no one ever addressed the question of whether those pericapillary things are actually separate tiny little pockets. But what we're showing is it's all connected throughout the body.
So you could strip out everything but the interstitium in a human body, and you'd see a human body.
And we do this actually. It's called decellularization. And about 20 years ago, and I was involved in some of this because of the stem cell work, it became clear that you could digest all the cells out of an organ and then put new stem cells in, and they would repopulate and recreate the organ. And they were growing along the extracellular matrix, which is mostly collagen, but there are lots of other biomolecules. And you could reconstitute an organ. And the idea of this was you could take, for example, a pig liver, which is about the size of a human liver, decellularize it to get rid of all the pig cells, and then put the cells of a patient who needed a liver transplant into that, and it would repopulate and create a human liver made of cells of the person who's going to receive it. That's sort of the pie in the sky idea. This has actually been done with urinary bladders
It's a
successfully.
bit like 3D printing using the design as the interstitium, right?
But what's happening is the cells are interacting
Using,
with each other like a
organic
complex
material.
system to self-organize around the structures, right?
Right. Yeah, so it's all about how complexity works. Neil, did you want to show any more slides, or should we take the
No,
share
I'm sorry.
screen off?
We can take that.
I just
So
want to see your
I have
face.
one comment,
Yeah.
and I'm reminded of when I was listening to your book, how you talk about the early microscopes discovered cells, and so everybody concluded that we were sort of cellular systems,
Right.
but that now more modern ways of observing led people to understand we are fluid. And I thought that was interesting. And then that and this reminds me yet again, this is my comment of what I learned as a knowledge worker at CIA is
I would love to be, maybe I'll just lie and tell people that. I was a knowledge worker at the
Well,
CIA.
yes, I'm going to use that as my intro now. I like it. But what I learned, but big takeaway of many about kind of working, trying to develop knowledge, is that everything we know is a function of our tools for knowing.
Yeah.
And every person, every generation makes the mistake of thinking that this set of tools that they have at the moment is like the final set of tools that's delivering wisdom, when in fact, you're always in a transition period from one set of tools to the next set of tools. And I just wanted to share that.
Well, and the endoscope, this new fancy endoscope.
Yes. Is what showed
I would, without them having that in their hands and paying attention to something novel and bringing it to me and saying, what is this? And the fact is that other groups had seen these spaces and reported on them, but they were all endoscopists who weren't interested in the science. And they all just showed it and said, well, that's there.
Right.
And no one else was reading those papers except endoscopists, because why would any scientist be interested? They weren't. So it's already there in the literature.
Yeah.
And going back, people are like, since we published the first paper and it's accelerated since, people are like, well, so-and-so saw this in 1943, so-and-so. And in our first paper, there are publications where we found a handful where clearly people saw this,
Yeah.
but they had no context,
Right.
or no one noticed that they had said it because it didn't fit